So good morning students. In the last class we had discussed about the endosperm. Okay, there are three types of endosperm formation. So today we will study about the embryo. See, first of all, in double fertilization, you had already studied about how embryo is going to form. Okay, that one egg cell that is haploid fused with one male gamete, which is also haploid, and after fusion of this one egg cell, which is haploid, with one male gamete, which is haploid, it will form a zygote, which will become a diploid. And this zygote, after post-fertilization or the events which are going to take place after fertilization, so that zygote will convert into embryo. That zygote will convert into the embryo. So how this embryo is going to form, that we have to see. From the zygote, how this embryo is going to form, that we have to see. First of all, after fertilization, see, after fertilization, what is the result or what the product means, that is the zygote. So when the zygote is going to form, the zygote will undergo into a rest of period or period of rest. For some time, the zygote will undergo into the rest or it will take the rest, then grows and it gives a size to the embryo. Okay, after taking the rest, then it grows, then it gives the size of what? Size of embryo. So here, so see here, when this zygote is going to grow and that is going to form the embryo in monocot and dicot. You know the difference between the dicot and monocot. Monocotyledons and dicotyledons. Dicot means where there will be two cotyledons. Monocot means where there will be only one cotyledon. So here the development, the stages of are the early stages, also we can say the early stages of embryonic development or the embryo development, which is known as the, and that particular stages is known as the embryogeny. Embryogeny. So this early stages of the embryo development, it will be similar in both. It will be similar in both dicot and monocot. Whatever the early stages of the development of zygote, from the zygote to the embryo, whatever the early stages means first stages, initial stages, that stages will be similar in both dicot and monocot. So let's see what are the particular stages over here, and then we will see about the embryo of or dicot embryo and as well as the monocot embryo. So see here, first of all, I will explain you once again, that is the structure of ovule in which there is a presence of embryo sac. Here, These are the two synergids, one egg cell, and this is the polar nuclei, and these are the three antipodal cells. Okay, these are three antipodal cells. This is the polar nuclei. Polar nuclei fuses with the one male gamete, and it will give rise to the primary endosperm nucleus, which further it will develop into the endosperm, which is one of the nutritive tissue for whom? For the developing embryo. And here these are the two synergids, and this is the egg cell. Here the egg cell fuses with one male gamete, and it will form the zygote, and this zygote will develop into the embryo. So first we will see about the zygote. Already you know zygote is a single which will have a one nuclei. So for example, if we take this zygote, this is the zygote with the diploid nucleus, or this is one of the cell after fusion of egg cell with male gamete. Here it forms the zygote. This zygote will undergo into the mitotic division. Which division? That it is the mitotic division. So this zygote will, after undergoing into the mitotic division, here it will form a two-cell stage. Okay, two cell. This cell will divide. The zygote will divide, and it will form the two cells. And this stage we can call it as the pro-embryo, which is the first early stage of what? Of the embryo. That's why we are calling it as the pro-embryo. And these stages are similar in both monocot and the dicot. The next, what happens means, here one cell will form whatever cell which is present at the bottom region. It starts dividing transversely. What it will do? It will start dividing transversely and making a cell wall which is in the form of transverse. And you can see it is at here after some division. Here it is going to form. See, here the division is transverse. Then again here it is going to divide into a horizontal way and then into a transverse way. So in this form, many cells are going to divide and they will form a globule-like structure. What it is going to form? It is going to form a globule-like structure, and we can call it as the globular embryo. What it is? Next stage, after zygote is the first stage, pro-embryo is the second stage, third stage is the globular embryo. Okay, after globular embryo, here the cells. Here also the cells will divide first transversely, then horizontally, and here it will make a heart-shaped structure. Then a cell is going to divide repeatedly in this way, and this stage we can call it as the heart-shaped embryo. Okay, this is the fourth stage, that is the heart-shaped embryo. Okay, so which is, you can see the whatever the cells which lies at the bottom, First, it will form a globular-like structure that is known as the globular embryo. Then again it will divide. This whatever the cells which are present in these globules, again it will start repeatedly, and here it is going to form a heart-shaped structure, and that is known as the heart-shaped embryo. From one zygote to cell stage, that is the proembryo. Third stage, that is the globular embryo. Fourth, that is the heart-shaped embryo. The next, and these cells, whatever present as suspension, these cells we can call it as the suspensor. Okay, which are holding the bottom cells. Okay, so that cells we can call it as the suspensors. The next, after heart-shaped embryo, how? See, these all stages, these 4 stages are similar in both monocot and dicot. Now the difference will come in monocot and dicot. So first, how this heart-shaped embryo is going to convert into the dicot embryo. So that we will see here. Now let's see. This is the dicot embryo, and this dicot embryo is the mature embryo. So let's see what are the regions of this cell. Now listen here. This heart-shaped embryo gets matured, okay, and it will form a dicot embryo, or either if it is dicot, then it will convert into the dicot embryo. If it is monocot, then it will mature into the monocot embryo. So this heart-shaped embryo will give rise to a mature. Here I am taking an dicot embryo. This is the dicot embryo. So here what happens means the first cell will be like that only, and the suspensor is going to reduce, and the cells which are present in the heart-shaped structure, that will form two regions. What are the two regions? That is one is the hypocotyle. This region we can call it as the hypocotyle, and this region, this region is the epicotyle. Here what happens means one embryonic axis is going to pass. Okay, a typical dicot embryo consists of a embryonic axis. Okay, so that embryonic axis will differentiate all the parts. So see here, you can see this region. This is the embryonic axis, a space between the two cotyledons. This region is the embryonic axis. Okay, so this embryonic axis will divide this into two cotyledons. Okay, and the region where the cotyledons are present, this region is known as the epicotyle, and these two are the two cotyledons. Okay, that region is epicotyle, but these two parts are the what? That it is the cotyledon 1 and 2. This embryonic axis is going to make a division to form the two cotyledons. Then the upper portion, which is the cylindrical portion, just above this embryonic axis, and where the embryonic axis is going to end, this region is known as the plumule. This region is known as the plumule. Okay, where the embryonic axis it terminates. Okay, above this plumule, whatever the cylindrical region is there, that region is known as the hypocotyle region. Okay, and here some cells are there. That cells is forming a radical. What it is? That it is the radical. In a simple way, here I had drawn a dicot embryo. This is also one of the dicot embryo, where you can see clearly the different parts. So this is the hypocotyle region, and this is the epicotyle. See here, epicotyle, hypocotyle, and this area or this axis is the embryonal axis. This axis will divide into cotyledon 1 and cotyledon 2. That is the two cotyledons. Okay, cotyledons means that is the seed coat, which stores the reserve food material. Okay, the next here this end where the embryonal axis is going to end, that region is known as the plumule. This region is radical, which is covered with a root cap. Okay, so this is about the dicot embryo. Next we will see about the embryo of monocotyledons. This is about the embryo or the dicot embryo. Okay, next we will go for the monocot embryo. next that it is the monocot embryo. Here, the monocot embryo will possess or it possesses only one cotyledon. In last topic you had seen there will be two cotyledons, but here it possesses only one cotyledon. So let's see what is that cotyledon. Here, whatever in grass family, as you know, the grass in grass family, that grass is the one of the monocot plant. Okay, so in grass family, whatever the cotyledon will be there, only one cotyledon what I told, that cotyledon is called as, it is also or it is called as the scapula. See here, this is the structure of monocot embryo, which is one of the very important. Otherwise, they will ask monocot embryo or the grass embryo. The question will be asked: draw a neat label diagram of monocot embryo or the grass embryo. That means both are same. Okay. So here, first we will see. In this structure, there is an embryonic axis. Here, this region is the embryonic axis, which is forming only one cotyledon. How many? Only one cotyledon. And that cotyledon here is known as the scatulum. Scatulum, which is present at the one lateral side of the embryonic axis. That means this is the embryonic axis. One cotyledon is located at one side of the embryonic axis. Okay, and this region where the embryonic axis is going to form one cotyledon, this region we can call it as the epicotyle. Okay, this epicotyle where it is, this embryonic axis where it is going to terminate, and here this region, cylindrical portion, we can call it as the hypocotyle, where there is a presence of radicle. This region is the radicle, which is covered by the root cap, and these two things or the two parts, radicle and root cap, is covered in an undifferentiated sheath, and that sheath is known as the coleorhizae. Okay, radicle and root cap are covered by an undifferentiated sheath, and that sheath is known as the coleorhizae. And here in the epicotyle region, there is a presence of a leaf-like primordia. Okay, leaf-like primordia, many primordia will be there, and that is known as the shoot apex. You can see leaf-like primordia, and this leaf-like primordia are located in a hollow folia structure. This region is the hollow folia structure, and that hollow folia structure is known as the colloidal hollow foliar structure, in which there is a presence of many leaf-like primordia, and that is known as the shoot apex. Okay, epiblast, which is one of the covering of the monocot. So this is about what? Monocot embryo. Okay, many times they had asked in a theory exam, that is about the monocot embryo. Draw a label diagram of monocot embryo. The next topic in this chapter, that it is about the seed. So let's see about that one. Next topic, that it is the seed. Seeds. Here, in sexual or during sexual reproduction in flowering plant, what is the final product? Means that is the seed, or we can say in angiosperm, seed is the final product. Seed is the final product of sexual reproduction in angiosperms. It is the final. At last, after pre-fertilization, fertilization, and post-fertilization, what is the final product? Means that is the seed. In whom? In angiosperms. So what happens means during this process, first the zygote is going to form, then embryo, then the seed is going to form. So here, the seeds are, where do you found the seeds? Means seeds are formed inside the fruit. Inside the fruit. Where do you found the seeds? Means that is inside the fruit. Okay. So you recall the structure of ovule. Ovule will change or develop into the seed, and ovary will develop into the fruit. So these seeds are present in the inside the fruit. And here, as you know, see, for zygote will develop into the embryo, and primary endosperm nucleus will develop into the endosperm. Okay, here endosperm is one of the nutritive tissue, okay, which is helping or which is developing this embryo. Embryo means that will further it will form the seed. That means here endosperm is providing a nutrients to the whom? To the developing embryo. That embryo will convert into the or develop into the seed. See, a typical seed, a seed will consist of a, this is the seed. Okay, if you see the lateral section of the seed, there will be presence. If you talk about the dicot seed, here there will be presence of a two cotyledons. This is the one and this is the two. These are the two cotyledons. Okay, two cotyledons, or else we can also call it as the seed coat. Cotyledons, which are also known as the seed coat. What is the function? What is the role of these cotyledons? Means these cotyledons will store the food material. Whatever the food source will be there in the seeds, that is present in these cotyledons. Okay. Then here it is consist of a seed coat, cotyledons, and an axis. This is the axis, and this axis is known as the embryonal axis. And here in this embryonal axis, a short plumule will be there, which will give rise to, after germination, which will give rise to the stem. Plumule, and at the bottom there is a short region or a short part that will be known as the radicle. Okay, radicle, plumule, two cotyledons will be there. So these are present in the simple seeds or the of the embryo of the simple structures. So here these two cotyledons are the thick swollen parts of the seed, which we are calling it as the cotyledon. See, mature seeds, whatever the seeds will be there, that mature seeds, it's of two types. What are the two types? Means that is the non-albuminous, and one more, that is the albuminous seeds. What are that? Seeds are of two types. That is, one is non-albuminous and one more, that is the albuminous. Okay, these are the two types of seed. Here non-albuminous seeds will not have any residual endosperm. Endosperm is totally utilized by the developing embryo. That type of seeds are known as the non-albuminous. Means the seeds have no residual endosperm because the embryo is totally utilizing the endosperm. That type of seeds are known as the non-albuminous, as it is completely consumed. It is completely consumed during embryo development. Okay, and what do you mean by albuminous seed? Here there are some examples of this non-albuminous seed. That is the pea. Here I will write examples: pea, groundnut. These all are the pea and groundnut are the non-albuminous seed where there is no residual endosperm because the endosperm is completely consumed during the embryo development. Here, what do you mean by albuminous seed? Means where it retains a part of the endosperm. Okay, it is not completely used during the embryo development. Here, seeds will retain a part of endosperm. Okay, where it is not completely consumed during embryo development. Some part of the endosperm will be persist in these type of seeds, which are that is the albuminous seed. And what is that means? Examples are wheat, maize, barley, castor, coconut, etc. So this is about what? Albuminous seed. Okay. So this is about introduction of seeds and the two types of the seeds. So in the next class we will see some more information about the seeds. Okay. Thank you.